Good afternoon. My name is Joel Hewitt, and I'm a subject matter expert with the Homeland Defense and Security Information Analysis Center, or HDIAC. We are excited to host today's webinar on detecting unexploded ordnance through changes in plant health, and we thank you for your attendance. Today we have two presenters from Missouri University of Science and Technology, and we'll introduce them further here in just a bit. Also, note that we will be recording the webinar, and we will post a copy of the webcast and the slide deck to our website, hdiac.org, and both will be available for download there tomorrow. And as a short prefatory note, the chat function in the top right-hand corner of your screen is enabled. So please feel free to type in any questions you may have as we go along, and we will collate those questions and answer them and any others during a question and answer session at the end. Finally, I'll share with you a bit of background information on our center. We are a Department of Defense sponsored entity, one of 3 Information Analysis Centers, or IACs. Organizationally, we fall under the Defense Technical Information Center, known as DTIC, and the Assistant Secretary of Defense for Research and Engineering. Our mission is to be the go-to R&D, S&T, and RDT&E leader within the Homeland Defense and security community. We achieve this by providing timely and relevant information, superior technical solutions, and quality products to the DoD and HDS, COIs, and COPs. In doing so, we are able to help solve the most challenging technical problems facing the government. We pursue this mission across 8 focus areas: alternative energy, biometrics, CBRN defense, critical infrastructure protection, cultural studies, homeland defense and security, medical, and weapons of mass destruction. A critical tool for achieving this mission is our external subject matter expert network. Both of our presenters today are SMEs that we've recruited into membership. While HDIAC's network is quite extensive, we have a fairly small staff, so we do rely on the support of our partners and our external SMEs. So if you are interested in joining us and have expertise in one of our 8 focus areas, please visit our website and apply. Or please feel free to contact me directly. So what do plants and plant health have to do with the application of new and novel technologies to the detection of unexploded ordnance, including aging landmines? Well, first a note on terminology. Here we are using the phrase unexploded ordnance, UXO, as shorthand for the less recognizable and less wieldy term munitions and explosives of concern. This term encompasses UXO, but it also includes the categories of discarded military munitions and, importantly, munitions constituents. Efficiently detecting munitions and explosives of concern is an important capability for DoD to have, both to meet its missions at home and abroad. Directly identifying UXO threats in the battle space is an obvious concern. But also, both DoD and a host of federal civilian agencies are responsible for managing public lands that are known already to contain harmful munitions or their constituents. For example, the Bureau of Land Management, or BLM, which is part of the Department of the Interior, oversees about 5 million acres of land that is currently open to the public that may contain munitions and or explosives of concern. So for a sense of scale, that's an area approximately the size of Massachusetts. Since 2001, both DoD at large and the individual service branches have been directed by civilian leaders to implement what are called Military Munitions Response Programs, or MMRPs. With these, DoD is charged with identifying the location and the concentration of any UXO and UXO constituents that may be there. They are also charged with setting remediation priorities, timelines, and then cleaning up contaminated areas to meet federal standards. So to effectively do this, there's a clear need for next generation technologies for use as wide area assessment tools to identify and delineate areas that may be in question. Airborne LiDAR, orthophotography, infrared sensing, magnetometers, electromagnetic induction sensors, and other means can assess thousands of acres per day. But many of these methods are either limited or they're hindered by the presence of vegetation or biomass. Thus, today's discussion surveys an emerging use of hyperspectral imaging technology that could help reverse this equation, capitalizing on the natural stress response of contaminated plants to identify munitions or explosives of concern. And so this kind of advance could open the door to several near-term discrete applications. For example, using hyperspectral phytoforensics as a supplement or an add-on to conventional detection methods. Or more broadly, this could lead to future applications including assessing plant life from the air to detect a whole host of variables. Now to introduce our presenters. Our main speaker today is Paul Manley, a doctoral student in civil engineering at Missouri S&T. He began his work with explosives and remote sensing while at the Virginia Commonwealth University, where he received his master's degree in biology. At Missouri S&T, he has integrated the use of remote sensing technologies into a lab that is primarily focused on plant-level uptake of contaminants. Secondly, Dr. Joel Burken is the Curator's Distinguished Professor and Chair of the Department of Civil, Architectural, and Environmental Engineering at Missouri S&T. He is also the Director of the Environmental Research Center for Emerging Contaminants at the university. And I hope I pronounced your name correctly. We'll see that in just a second. And with that, we turn it over to our two presenters.
Thank you very much for the introduction.
Thank you.
We're glad to be here today and would welcome any questions or input as we go through our topics. And we'll go right to our slideshow here. And basically the overview that we're looking at today is how to utilize plants in assessing chemical signals in the environment around us and primarily in the subsurface. And recently we switched this work to looking at explosives aspects and directly looking at agricultural species and their drought and how the response to explosives is distinct from that of the stress of explosives exposure. We transferred that into hyperspectral imaging, and we found the ways to discern between the natural and the anthropogenic stress that the explosives can cause. The initial work demonstrated the potential in scientifically for advanced technologies related to chemical delineation in the environment, particularly plant exposure to explosives, and then transitioning to the capability to do remote sensing for subsurface UXOs that are exposed with the explosive compounds detected previously. The research potential is very impactful for DoD missions, looking at the capabilities to assess vegetation, which is normally thought of as an interference as a way to detect UXO, but also to look at explosives and other environmental pollutants or environmental compounds that are in the environment around us.
So just to introduce this idea as an issue, a global issue, this map is showing you mine contamination as of October 2013. So you can see where landmines have, specifically another UXO, have been an issue across the world in South America, Africa, etc. So what you'll see later is that we take this information, and since we're using vegetation, we kind of try and put two and two together and see where this research is most effective, potentially.
So the foundation of our work and where we kind of started was to look at exposure to environmental pollutants, essentially the health burden on the human body due to contaminants in the environment around us, and understanding that plants occupy the same profile of our biosphere as we do, and that they're exposed to the same compounds. And if you consider plants as really a master of mass transfer, They harvest all they need from the subsurface and from the atmosphere around them, and they're place bound. So if there are chemical molecules, essentially pollutant molecules in that environment, plants are very efficient at accumulating those molecules, and then looking at advanced techniques, we can actually assess those concentrations in the plant. And then in the work we're presenting today, how can we transition that into understanding the stress caused by these pollutant molecules on the plant and discerning that plant stress as either being due to natural sources of drought or low nutrients versus the chemical exposure that we see from the surrounding air, water, direct contact, or from the soil, and this also has implications related to food security. The primary methods we've developed to assess chemical exposure to plants was initially developed by Don Wroblewski with USGS. The fundamental concept is to take a tree core or a tissue sample of the plant in some way and bring that back to the laboratory where you can do a very distinct chemical analysis to find the molecules of interest, the pollutants of interest. And by understanding the coefficients of transfer into the plant and partitioning within the plant, we then can back calculate what the environmental exposure could be, such as the subsurface concentration in the soil, groundwater, or vapor phase of the vadose zone. The next step in our understanding is really determining which compounds can get into a plant system, either by transfer from the atmosphere, but primarily through the root membranes. And by understanding that transfer, we can see which molecules from unexploded ordnance or from any environmental source are likely to transfer into the plant. And by understanding that transfer as a function of compound properties, we can then predict which molecules we would see in the plant, and therefore which exposure could lead to stress within the plant, again due to the anthropogenic compounds that are in the surroundings. So that brings us to our new methodology. Once we understand which compounds can get into a plant and how efficiently they're transferred, then we can look at new methodologies to assess the stress that these compounds may cause on the plant systems.
So just to give you a quick overview of the different sensors that we're talking about here, obviously there are RGB sensors similar to what you see in your phone. These kind of cameras can be mounted to UAVs where you can visibly assess plant health. You can also do some sort of like structure for motion and look at biomass. There are also multispectral sensors that you can fit to drones. We deal specifically with Hyperspectral. That is a headwall nano hyperspec. We'll get into the details of that later, but this one it's a versatile camera because we can use it in the greenhouse as well as mounted there to our DJI M600 Pro. With that, application in the field, we have to have an inertial measurement unit. That's that orange box on the side. The camera is a line scanner. It's not a regular point and shoot, so it takes about 1 pixel by 640 pixels, and as it moves along its flight line, the IMU data is integral to stitching all of those pixels together at the end during the orthorectification process. And kind of continue on with the different sensors. So RGB, we're talking about 400 to 700 nanometers roughly in the visible range. Multispec, depending on your camera, in the visible and also out into the near infrared, but 8 bands, maybe a few more, camera dependent, of course. Our hyperspectral camera deals in the similar range from 400 to 1,000 nanometers with 274 bands in between. So what this looks like in the software, we use NV specifically for image analysis, is with RGB you get 3 bands, right? Red, green, blue. Low spectral resolution, you see a peak in the green there from the plant. When you add in more bands out to the near infrared, you can start to see what looks like a typical vegetation reflectance spectra, but still there are plenty of gaps in between those points. And when you add in, again, 274 bands, those gaps are filled in, and you can look at high spectral resolution detail of plant health. And so, hyperspectral imaging has been used for many years. It's been used in mineral identification. There are even food safety applications where you can scan some produce or some sort of meat and see if there are any contaminants on it. We use it specifically here for drought and chemical purposes. And one of the metrics that we use to assess plant health are called reflectance indices. And you don't have to be concerned with the abbreviations there, but more so what they tell you about plant health. So they are physiological and morphological metrics that deal with photosynthesis, stress, biomass, pigments, etc. Generally, a reflectance index is a simple algorithm. It's just the difference of two wavelengths divided by the sum. There are simple ratios and other more complicated equations used for these, but most of the ones used in this study follow this format here. And two of the more widely known ones are the photochemical reflectance index, which is a pigment index related to photosynthesis, and the normalized difference vegetation index, which is a biomass index. So as was mentioned earlier, this all kind of started at Virginia Commonwealth, at least for me, at Virginia Commonwealth University. Before I started my master's, there was an Army Corps grant. My lab was approached to do some hyperspectral analysis with a Field Spec Pro and some other physiological parameters to relate to that spectra and indices. And so if you are interested, look up Julie Nauman or Julie Zinnert. It's the same person. But yeah, so you can check out these and other papers that really where all this started and what brought me out to Missouri S&T. So again, back to this map, you can see where there have been issues with UXOs. In terms of landmines, there have been about 110 million planted since the 1970s. This is an estimate from Walsh and Walsh from 2003. And so my research focuses on using plants to detect UXOs and landmines. So if you overlay global vegetation cover, you can see where this research is potentially applicable in Western South America, Sub-Saharan Africa, Southeast Asia, and there are even parts of Europe. Currently, what is going on for detection, they have people out in minefields with detectors walking around. They also have animals on leashes that they lead around to try and sniff explosives. There are heavy equipment that till up the topsoil to just detonate them at the same time, and ground penetrating radar units where they actually try and sense them in the ground. The thing with these methods is that they all involve people down in the minefield, close proximity to the danger, and the vegetation is considered an issue. So what we're proposing here is to increase that distance to the explosive using remote sensing methods. And by doing so, we're also getting people out of the minefield, and we're also increasing the area that can be covered in the same amount of time. And the way this works is you have a UXO in the soil that's been in an area that was raised to the ground and allowed to revegetate, or was originally vegetated. And over time it will come into contact with moisture in the soil or from rain, and that casing will break down and let the compounds leach out. So here you see a plant that's been exposed to explosives, a healthy plant, and the light's coming down, it's reflected off, your sensor flies over, whether it's on a UAV or a manned aircraft, and you get a reflectance spectra similar to this. So what you're seeing here is low reflectance in the blue and red wavelengths from pigment absorption, a peak in the green around 550, that's the green that we see from the plant, and as you transition from the visible to the near infrared wavelengths, that region represents biomass, and that's from the high scattering within the internal parts of the leaves. So if you look at, this is Comp B actually from some previous research, so Comp B is a mixture of TNT and RDX. So if you look at this spectra, it looks very similar. However, when you overlay the two, you can start to track changes in how these specific wavelengths respond to the presence of explosives. What we did here at S&T was in conjunction with the drought study we were working on. We used the same agricultural species, 2 types of maize and 1 of sorghum. There were 8 replicates for each group. And for our treatments, we had a drought group and a group that was exposed to RDX. We calculated the the field capacity of our soil, and we held our controls at 90% field capacity, our drought group at 60% of that field capacity. And for our explosives, we used 250 milligrams per kilogram of RDX. And we actually germinated those groups separately and then transplanted them into the explosive soil. And the reason we did that is because we didn't want any germination effects on the plants. We were looking more at how the contaminant affects the reflectance. Because what happens is we obtain the material and we mix it into acetone, and then that solution is then mixed into soils, and we allow the acetone to evaporate, leaving behind the explosives. So again, we have the Headwall Nano Hyperspec, which gives you a spectral range of 400 to 1,000 nanometers. In the greenhouse, we use the halogen light source. This is to mimic full sunlight. You get all the wavelengths that you need off of this light source. And then the Headwall software actually converts the raw images, which are digital numbers, to radiance, and then those images are what are used in ENVI. And I wrote a code that takes all of these images. After you have 8 replicates of 3 different groups, 3 different species, the number of images starts to add up. So I wrote a code that handles these images, so I didn't have to do it all by hand, and I'll show you more about that in a minute. So to give you an idea of what the plants look like after 4 weeks of treatment, you can see that the drought-susceptible maize, the controls look nice and healthy. The drought group, not so much reduced. Visibly, you can see the reduced biomass, and the RDX group had leaf curling at the edges and also necrotic regions. With the drought-resistant maize, you can see that the drought group looks pretty good, just a visual assessment. The RDX, however, did not take to that too well. And with the sorghum, similar responses here, slightly reduced biomass from the drought group, and in the RDX, it's more the leaf curling and necrotic regions. So the code that I wrote for this project, it takes in these radiometrically corrected images. It then puts it runs an index on each one, and this is the difference vegetation index. So it highlights the vegetation and it mutes the background, and then it takes everything above a certain point as a threshold and builds a mask from that and applies it to the original image. So now we're just left with pixels that are plant, and we don't have any of the background interference. And then 18 reflectance indices are run on those images, and those average values are output to a text file where you can then do whatever statistics you want on them in SPSS, R, whatever you use. So some of these indices I'd like to highlight. The first one here is the anthocyanin reflectance index. It uses pigment response to discern stress. So with higher values, you would expect to see a stressed plant. It's also used for new growth, but in the case of these plants, there was no new growth after 4 weeks. So it's just we're just talking about stress here. So this one is kind of all over the place. Not really getting some of the responses we would expect from RDX exposure. With NDVI, this is a normalized difference vegetation index. The lower values would indicate reductions in biomass, and this is more of what we would expect to see from exposure to RDX. And PRI, or the photosynthesis index, also kind of what we would expect to see as RDX gets in the plant and inhibits photosynthesis. So when you put these into a principal components analysis, those index values for all plants, all treatments, you can see that the RDX group is completely separating out from the drought and controls, and the percent variance of each axis is written out there. So with all treatments and groups, we're looking at 67% of axis 1, the variance explained there. When you remove RDX, you're seeing some grouping, but no real separation between the drought and controls. And then looking at control and RDX alone, again, more separation and grouping. What's really interesting on this slide, though, is the drought and RDX. There's complete, these ovals have been shifted, but there's complete separation and grouping between a natural stress, drought, and anthropogenic stress, RDX. So this is really promising for future work and being able to discern between those two types of stressors and limiting the amount of false positives or negatives we may see in the field. I wrote another code that extracts all the spectra for each plant, and then this is an average for each group, each treatment, I should say. So for the drought-susceptible corn, what you're seeing here is that dashed line that's higher up in the visible. That's the RDX group, and where the lines are red, they're significantly different. The wavelength responses are significantly different from controls, or the dotted line. And then the gray hatched area is where the drought and RDX wavelengths are different. And while we don't have any pigment measurements, what we're seeing here is that the RDX group is actually reflecting more light in the visible portion, and we know from previous research that RDX can cause reductions in pigment concentrations in certain species. So what we're seeing here is increased reflectance due to possibly a reduction in pigment concentrations. And that steep slope around 700 nanometers, that's what's called the red edge, and we're seeing there a blue shift or higher reflectance earlier in the RDX, which is also a stress indicator. And as you transition over into the near infrared, we're seeing reductions in biomass in both drought and RDX groups. And a similar response here from the drought-tolerant maize. Sorghum responded a little differently in the near infrared, but very similarly in the visible wavelengths here in terms of the RDX group. So these are our first derivatives of those spectra, and what I'd like to use these for is to identify wavelengths that have different responses based on treatment, and these will be important in future index development, because what we would like to do is develop our own explosive specific index and potentially even come up with new drought indices. And so you can see where some of these could be good potential indicators of specific wavelengths that are affected by explosives. So in conclusion, the ag species that we did use, the maize and sorghum, did respond differently to our treatments, and that hyperspectral imaging was effective in discerning between those two treatments, as well as from healthy plants. And of course, plants are useful tools in exploring what is in the subsurface. As Joel mentioned earlier, they're already there, they can't move, but somebody else could have put something in the ground, whether it's a UXO or some other sort of compound, and these plants are anchored there and can be used as phytoforensics tools in the detection of those. And so what we'd like to do now is strengthen the link between, where the contaminant is, if it's leaking out, how quickly it goes from leaking to plant uptake and plant exposure, how that affects the plant's hyperspectral response and other physiological parameters as well, and how can we tie that into flying a UAV or an airplane over an area and picking out these hotspots. So in the future, one way we'd like to do this is by broadening the species. start looking at more native plants and not so much agricultural plants. We also want to broaden our stressors, so include TNT and HMX. HMX is similar in structure and mobility to RDX, and within this we also want to include nutrient deficiencies. And scale this up. This summer we have plans to use mesocosms and we'll contaminate the soil there, put some plants in it, fly over it with our UAV and camera. That's it right there, actually. And then quantify the uptake. So a method was developed here where you can take a plant tissue sample, freeze it, grind it up, centrifuge it, and then extract the liquid and run that through liquid chromatography with tandem mass spectrometry, so we can actually start linking the amount of explosives that have been taken up to the physiological parameters and hyperspectral imaging. And so yes, we recently acquired a fluorometer, so we can look at fluorescence and how that responds to explosives uptake, as well as stomatal conductance. We just got a pyrometer, so we can do those two things and do some other pigment analysis as well. And then again, target the wavelengths, any wavelengths that specifically respond to explosives, and that goes into our index development.
Thank you, Paul and Joel, very much. So the need to detect unexploded munitions is, of course, not a new one. It seems about once a month in Western Europe, a construction project will uncover either an artillery round or an air-dropped bomb from either the First or Second World War that failed to trigger. I know in February and then again in April, they discovered a 1,100-pound bomb both in East London and in downtown Berlin. So as a result, many of the standard detection methods in use today rely on fairly mature technologies. And even so, some of these methods can be limited in two important ways. First, like the electromagnetic towed array system that's pictured here, and this was used at the New Boston Air Force Station. These systems are most effective when they're deployed on the ground, as Paul said. This is especially true when trying to detect UXO proper. So any land-based approach is almost necessarily a slow, inefficient, and sometimes cumbersome process. It is not an ideal or really even a workable solution for, say, a target scope area of 5 million acres. Secondly, the presence of vegetation can limit the usefulness of some of these methods to detect both munitions and explosive chemicals of concern. Infrared sensing and the use of ground-penetrating radar can be particularly affected by the presence of vegetation. In fact, infrared sensing is most effective on terrain that is entirely barren. So what the hyperspectral imaging-based technology discussed today, along with the indices and the code that Joel and Paul have put together, what this offers is essentially the ability to tap into the work or the automatic stress response that contaminated plants are already sustaining or undergoing. So in many senses, this allows for a more direct detection of the presence of munitions constituents or other chemical, perhaps even biological, contaminants. This could aid both DoD and federal land managers in assessing target areas beyond just the few that are marked as highest priority for remediation. The application of hyperspectral imaging to biomass or plant matter could further open up a wide array of potential innovative uses. This includes long-term environmental monitoring, essentially rerunning this process over a period of months or years, which is itself a requirement of DoD and federal agencies in munitions site remediation. And on a very broad level, being able to remotely monitor and discern whether plant stress is either naturally caused or anthropogenic in its source could lead to as of yet unknown military uses. And so now we will open it back up with Paul and Joel and answer any questions that have already come in. And please feel free to add in any questions as we go.
We can go ahead and start with the questions that were already posted, if that would be preferred. And the first question that was posted is, what are the plant species in the photo?
Yeah, so yeah, two types of corn and sorghum.
Yep. And then noted there, but the corn, there were two different types. One was drought tolerant and one was the native species.
As far as that light intensity question, yeah, in the future we're going to use a light diffuser to kind of knock down some of the intensity on the top portions of the leaves.
And the single plant variances, we did the analysis and did the statistics on all the plants exposed within a single group. We didn't have any direct concentration measurements in these plants that were presented here today. And the work going forward, now that we do have the signals and we have the hyperspectral imaging and indices that are of interest, we are going to be looking at doing direct concentration profiling of plants that are exposed and seeing if there's a more direct correlation between the in-plant concentrations is determined chemically and also the response of the plant as looked at through the hyperspectral indices. And we do assume that the RDX, being a triazine, but now with the toxicity of atrazine, does have that photosynthesis impact, and that could be why we saw the direct signal in a few of the different indices that related to photosynthesis. The next question is, have we looked at algorithms to identify plant defense compounds such as antioxidants or P450 that could be induced due to the exposure to the antagonist? No, we have not. We have not looked at secondary enzymatic response of the plant. There have been some studies that have looked at that, but we're looking at what are the hyperspectrals or remote sensing signals that we could pick up. And if we do have a very good tie to some of those direct measurements, hyperspectral, or excuse me, remote sensing and hyperspectral indices, we may look back at what is the cause of those, such as antioxidants, or what is the specific plant response.
So, through some of our previous research, the broadleaf species seem to respond to the treatment of explosives versus like nutsedge, which I used in an experiment a while back. With something like nutsedge, a monocot was not affected at all from any metric we threw at it. Elm and grape where they actually dropped leaves and regrew after being exposed to Comp B. Here, corn is also monocot, but it was affected. So, but it was just RDX here. So we are seeing species-specific and explosive-specific responses.
And another question is, how do we distinguish between atrazine and RDX? We have no indication that there is a difference at this point. We would look at what the specific responses are for RDX primarily, and then compare that to known responses for other antagonists, herbicides, pesticides, to see if there's an overlap. But we would be interested to look long-term at how precise can we be at many of the stress responses of the plants. Because most certainly what we're picking up could be induced by other chemicals, but at that point we could at least screen for the presence or absence of the stress and then discern, is this an area where there's likely an atrazine contamination perhaps, or if it's an area where unexploded ordnances or RDX contamination would be expected. And also look at the patterns. If we see a pattern of stress in the plants looking spatially, not on the individual plant, but over a large field or space, then we could better discern what the cause of that stress may be before actually going into the site and looking for direct assessment of plant concentrations chemically.
Yeah, I think the difference between is it atrazine or is it RDX, would be dependent upon where exactly you're sensing. And if you know that something has been produced there, tested, planted, whatever it may be.
Yeah. And the next question came in of what about the idea of seeding, potential UXO contaminated region? And I assume this is getting at, would we be interested in working with an area where we could do directed RDX contamination on a site and then see if we can pick that up in the stress? We'd definitely be interested in that field application.
Yes.
And that would be the next step in our progress.
Where we are, we're still so early on in this research that we've wanted to ensure that homogeneous mixtures in the soil to make sure that the plants are getting exposed. But in the future, we'd like to either work in these contaminated areas, or also when we scale up our research, plant some piece of explosives in a certain portion of the soil in a controlled environment, of course, and then see how that material moves through the soil, how the plants affect. One idea Joel has had in the past was, what happens if we put a chunk of RDX on this side of the tree? Will we see the response on that side of the tree, whether it's through tree coring or hyperspectral imaging, or will it move throughout? So we have all these ideas and things we'd like to pursue in the future. And yes, in regards to TNT, my previous research involved Comp B, which was the 60-40 mixture of RDX and TNT and RDX. And so with TNT, what you expect is it's not as mobile in soil or in plants. So what happens is TNT crosses the root membranes and tends to stay there in the roots. There's some evidence to suggest it can metabolize and send conjugates into aerial portions, but for the most part, if you were to expose a plant to Comp B and do some phosphor imaging, which the name of this person that did it I can't remember. Maybe Joel should know the phosphor imaging of TNT and RDX. I can get that reference if anybody's interested. Yeah. But you can actually see the compounds of TNT in the roots, none in the leaves, and RDX is just in leaves and not in the roots.
And that's one where the understanding of the transport in the plant is critically important. TNT does not transport very well. So any response we would see at the leaf level would probably be a stress induced by toxicity at the root level. Mm-hmm. And then what is the effect on the plant versus on the aerial tissue versus what is the tissue specific interaction, such as RDX impacting the photosynthesis pathway.
Right, because TNT there are papers out there that show that TNT will affect plant physiology, but we just won't see that response in the leaves necessarily.
We are also interested in what's the phenotypic response. So we're looking as well at how plants grow differently underneath the exposure to different compounds, not just a hyperspectral or reflectance imaging, but looking at what's the leaf area index, or is the branching different, or the angle of the leaf set.
Number of leaves.
And we could look at those as a more direct measure to get an aerial response with the hyperspectral or other imaging, and then get a more ground-specific response so we can understand that better.
We can still take questions as they're coming in. To Paul and Joel, I wanted to ask, what is the variables like, and have you looked at detecting the same chemical within the same species of plants at different ages or stages in its development?
So for this experiment and previous ones, I image on a weekly basis. So we do have that data. And, with explosives, Joel's previous research will show that the plants will actually take in the explosives within hours, less than a day, from time of exposure. So I do have the data that was taken over 4 weeks, imaging once a week. We could increase that imaging time and go through that data and see how the plants are responding. But for the purpose of presentations and whatnot, I have just always focused on the last week of data.
Mm-hmm. yeah, the more mature plants are of interest to us, but looking back at that is something we hope to do.
All right. Well, we don't see any questions coming in right now, but we are happy to answer them if one strikes one of our attendees later. Oh, we've got one coming in.
Yeah, and you know, like you said, with the age of plants, you know, obviously larger plants have more capacity to move more water and thus more contaminants. So you can see those. You can actually like quantify that based on the size of the plant.
Michael asked, have we looked at the bare spots of soil between fields and see if that might change the response? Our current techniques actually mask any of the soil from the image analysis. We certainly could look at patterns of the bare soil, and that's one of our hopes down the road, is to look at patches where there's actually mortality, and essentially the plants have been impacted to the level of death as far as being part of the patterning. So if we could see that bare soil spot surrounded by stressed plants, it's a high indication that there may be a source at that very point.
Well, as we wait for any other potential questions that may come in, I just want to mention a couple of services that HDIAC offers. If you have a more involved question that comes out of today's webinar, you can submit what we call a technical inquiry. You can go online to do this or contact one of us directly. We provide up to 4 hours of free research services in answering such an inquiry, provided it falls within our focus areas, and these have a fairly rapid turnaround time. Now, if your question or project exceeds the scope of our 4 hours, you can utilize our Core Analysis Task, or CAT. This is how we support research and development within the defense and federal communities. It's pre-competed and it's pre-awarded, and thus we can begin work on a CAT in as little as 6 weeks after a statement of work is signed and approved. And it does look like we'll have another question coming in. And just a note to anyone who, again, if you get a question later on today, please feel free to email either our presenters directly or me at HDIAC. We can answer those in due time.
And we'd certainly like to thank you, Joel, and all the group at HDIAC at the work at putting this together. It's been excellent working with you.
Yeah, it's been fun. It's been interesting, and we're looking forward to any future work that could come out of this.
Good deal. Here we go, another question. I'll quickly answer that last one. We will be posting both the charts, all these slides, and the webcast of the webinar on our website, hdiac.org, sometime tomorrow.
Yes, we would certainly be interested in surveying a munitions production facility. We've done the chemical analysis on a couple different areas where we've mapped out pollutants in the subsurface just from the plant sampling. But we have not been proposed with the opportunity to go to a site where there's a known contamination to see if we could pick that stress up either in the population dynamics or in the reflective indices of the vegetation on the site.
Daniel, thank you for your question, and we will contact you directly after the webinar as well.
Certainly. And again, if there's any other questions, please feel free to follow up with HDIAC and the group, and we look forward to those.
Yeah, thank you all for coming today.
Asking Daniel, yes, we have done perchlorate as far as the chemical analysis in plants. We have not to date done perchlorate for a reflective and reflectance impacts.
Mm-hmm. So we're definitely interested in adding in as many groups as we can, try and differentiate between these different stressors.
Final note, although the webinar will be available online tomorrow, you can actually go ahead and download all the slides here today. On the bottom right hand, it says click to download content. It'll be right there. Well, and with that, Paul and Joel, thank you again, and thank you to everyone for attending.
Thanks.
Thank you, Joel.